Hi everyone! Today I'll be talking about my project that I've been working on throughout this past semester, where I'm exploring the microbiome variations in honey from different ecosystems. To give you some context, this is a continuation of an independent project I started in 10th grade, where I looked at honey from beehives around New York City. I worked with a beekeeper to collect honey samples from around the city and with Cold Spring Harbor Lab to extract and sequence the DNA. I then wrote up a paper on my results and published it on bioRxiv. I love this project so much that I want to continue it and expand my samples to locations around the world with vastly different ecosystems. Although the samples from New York were distinct, it was not drastic as they all encountered relatively the same environmental factors. Based on some questions I developed from my previous study, I started a year-long independent study to look at the DNA profiles found in the honey from samples all around the world, with my main focus on trying to answer, how do external factors affect the microbiome of honey? Bees act as bioindicators because they pick up contaminants and debris from their surroundings and bring it back to the hive where it is incorporated into the honey. This turns the honey into a biomarker for bee health. Bees are dying at a rapid rate, so it is important to learn what they are coming into contact with. In order to understand why this information is helpful, it's good to know what makes bees so important. Bees are fundamental to sustaining our planet. They have co-evolved with the Earth, and with climate change, bees are unable to catch up to the Earth's current pace. Bees pollinate around 1/3 of the food we depend on and make 6,000 tons of honey. They also contribute over $400 million to the economy, making them an essential resource. Unfortunately, bees are dying at an alarming rate. This decline is known as colony collapse disorder, also known as CCD. CCD is the broad term given to a number of factors, one of which is climate change. I remember the first time I saw a rooftop hive. It was winter, and I walked outside to see hundreds of dead bees littering every inch of the ground. The problem is that with the warmer temperatures, thinking winter is over, the bees will leave the safety of their hives in search of food. This leads them dying either of starvation or freezing when the temperature drops. This decimates their populations. Other causes of CCD include habitat destruction, the rise of monoculture farming, and the increase in diseases and pesticide use. As you can see from this infographic, looking at the population of United States bees in 1947, there were 6 million hives. In 1980, that was reduced to 4.5 million. And in 2008, there were only 2.4 million hives remaining, and the numbers have decreased since then. Here you can see a chart created by the Convention of Biological Diversity in 2018, which shows the top 3 causes of bee decline in different regions of the world. While it's primarily the same factors, varroa destructors and pesticide use, these contributors are in different degrees of magnitude in various regions. The order of events that leads to bee decline can be summed up like so. Environmental factors such as loss of habitat can lead to bees having a compromised immune system. This creates an imbalance in the microbes. This is not a linear relationship, but more of a network with many other factors at play. An example of this is that an unbalanced microbiome can also lead to a compromised immune system. Ultimately, this leads to bee population decline and can cause food shortage affecting humans. So, what is harmful to bees? This image shows some of the culprits to the bee decline. In an article entitled "Bees Brought to Their Knees: Microbes Affecting Honeybee Health," researchers have outlined the specific organisms that are detrimental to honeybee populations. One of those is the deadly parasite Varroa destructor, or Varroa mite. The Varroa destructor is one of the leading causes of bee deaths and has traveled throughout the world, starting in 1904 in Indonesia and moving all the way across until most recently it has made its way back around to Australia. This graph has not been updated to reflect the evidence of mites in Australia in 2019. There is variance in different ecosystems for how long it took the mite to spread. The Varroa mite is a parasite that latches onto the bee and sucks their fat, as well as transmits a neurotoxin, an act made easier when the bee already has a compromised immune system, these hurting their effort to fight pathogens and parasites. These infections change the bee's microbiome not only within the bee, but also within the hive as a whole. Bees are very social, so it is very easy for them to spread diseases from hive to hive. As I mentioned before, bees act as bioindicators, giving us information on the hives and their location's health. When pesticides are introduced to a hive, they can cause pathogens directly, but they can also suppress the bee's immune system, making it easier for opportunistic pathogens to infect. The chemicals in the pesticides, as well as the microorganisms associated with them, are easily transmitted from hive to hive through a process called drifting, where an infected bee will try to enter its own hive but will get rejected. So it will have to find a new hive where it is accepted and is then able to infect and take down the entire hive. These pesticides are so toxic that they hurt not only honeybee health, but human health as well. One of the most dangerous family of pesticides are the neonicotinoids, which have a caffeine or nicotine-like effect in the bees. They will become addicted to it and keep returning to the same crops and become infected and die. Bees use dances to communicate pollen sources, so it will lead other bees to the same source, where they too will become addicted. Here we have a study that looks at the reaction that bees exhibited in the UK, Hungary, and Germany when they were introduced to neonicotinoids. As you can see in Hungary, the colonies are 24% smaller the year after exposure. The bumblebees had fewer queens, and solitary bees produced fewer eggs, all factors that would reduce the population size. The reason that the countries had specific reactions suggests that it's not just the neonicotinoids, but there were other factors that mediate these adverse effects, such as different crops in the area, varying ecosystems, different types of bees that populate the area, or duration of the presence of varroa mites. This slide shows the scientific evidence for why these pesticides are so bad, but what impact do humans have on this? Well, currently, in the US, there is no federal regulation on the deadly neonicotinoids, and they leave it up to the states to decide. While some have put in place suggestions on handling to keep people safe from pesticides, they don't take into account the bees or the environmental repercussions. However, on the opposite end of the spectrum, we have the EU, which has a total ban on neonicotinoids, except in enclosed greenhouses. Somewhere in the middle, we find Canada, which has strict regulations that allow for their use with the intention of eventually being phased out. Here's an image that I pulled from my in-depth literature review, sourced from the United States Ecological Survey of 2017. The image shows the estimated agricultural use of a specific type of neonicotinoid in 2011. The key shows the estimated use of agricultural land in pounds per square miles. As you can see, it is distinctly darker here in the South, in the Midwest, and in these areas on the West Coast. Over the course of my project, I've had difficulty finding clear answers to many of my questions. However, while researching, I also found this image, which was created by the University of Vermont in 2015. It shows the U.S. wild bees at risk with the scale showing dependency-weighted crop area by abundance on cropland. What do you notice? Well, if you're like me, then you saw how similar these graphs are, yet they are from two completely different sources and have different measurements. This exemplifies the direct relationship between neonicotinoid use and bee population decline. Created by the Dozel Lab at University of Champlain in Illinois, this image depicts the different ways that pesticides, viruses, and parasites, such as the varroa mite, are able to get into a hive and negatively impact the bees. Pesticide exposure can be traced to contact with pollen, nectar, or water, and pathogens can be transmitted through the food that bees eat, as well as several other methods. The consequences of infection from pesticides include changes in the gut bacteria and the heat shock proteins, as well as the impact on gene expression and receptor activity, to name a few. What we know is that neonicotinoids are very harmful to bees, not just by directly helping microorganisms that are hurtful, but indirectly by making it easier for the varroa mite to attack. The balance of the microbiomes is changed, and diseases are easily transmitted through a hive, from one hive to another. What is unknown is, however, is the different ecosystems create variation. It's not only this cause events, but also the response of the DNA profiles created in the honey, acting as a biomarker for the bee's health. Since we do know that pesticides are incredibly deadly to bees, scientists have looked for alternatives, such as biopesticides, which are made up of microbial pesticides, chemical pesticides, and plant incorporated protectants. The effect on bees is presented as non toxic. However, studies have shown that there are several that can affect the growth regulators in larvae, causing the bees to become deformed and unable to complete their roles in the hive, causing the hive to collapse. I have found a database with information on the ingredients, so once I get back my results, I can look for evidence of biopesticides. Now I'll go into my current study to try and answer some of the questions I have found. As I mentioned before, the top causes of bee decline is different depending on the region. While there is an overall trend of pesticides and varroa destructors being the leading contributors, they're in different orders, showing that depending on the ecosystem, it makes a difference, leading you to ask, how do bees' microbiomes vary across ecosystems? And can honey act as a biomarker to predict bee health? After a lot of research, my hypothesis has developed to be that the honey samples will be distinct from each other, with the samples from more remote locations having the most diversity and least amount of harmful microbes. Over the course of this project, I've gathered samples from across the globe and divided them into 3 groups based on the information that I've learned not only about the specific honey samples itself, but about its general location and what it looks like. First up, we have the green group, which contains samples that are unfiltered and unpasteurized, and they're the least likely to contain contaminants based on my hypothesis. The next is the red group, where it is more likely to have pesticides used surrounding the locations. And lastly, commercial. These samples are all store-bought and mass-produced, so they're most likely to contain evidence of contaminants. These are the specific microbes that I will be looking at once I get the DNA sequences back, and these have been shown to hurt honeybee health. I did not find them in my first study with urban samples. However, I do expect to find evidence in the red group of my samples where the locations are more rural. So far, as I mentioned, I collected my samples, and I've already conducted the DNA extraction using a DNA Easy kit. We have also obtained the primers to conduct the PCR, where the DNA present will be copied billions of times and to then send it off for sequencing. My overall goals of this project include to gain a better understanding of the threats that hives are facing, such as neonicotinoids and the role biopesticides play in causing bee population decline. In addition, I want to learn how honey can act as a biomarker for the surrounding environment and provide information on current bee health and the people who live in the nearby area. In my last study, I found a high concentration of cyanobacteria in one of my samples. Compared to the other samples I looked at, cyanobacteria has not only been proven to be detrimental to bee health, but also humans. One study showed that people who live in a close proximity to higher concentrations of cyanobacteria were more likely to develop ALS. I want to learn more about what honey and by extension bees can tell us about their environment, as they only travel about 1 to 2 miles from their hives. Lastly, I want to continue to hone my research skills and understanding of the process as I continue to pursue science. My progress to date includes a University of Reading microbiology class to gain a solid background. I also took a Codecademy Python class to learn how to model data. I've reviewed my previous study and asked questions and looked for places to expand. And I've conducted an in-depth literature review where I researched the answers to these questions and also found what questions don't have answers. I created a website to help people follow and keep up with my current work, and I've collected the samples with the help of family and friends. I've been able to collect my samples from across the world. And lastly, I've extracted the DNA. Once the samples have been sequenced, then it will be possible to try another method of primers known as indexing and see how they compare. We would also love to reach out to other labs. I would love to hear from others who have done similar work and perhaps share my own. Lastly, I would like to write up a paper about my results and, similar to my first study, publish it online. We have also looked into working with Oxford Nanopore, whose device would allow us to complete the lab work in much less time, cutting out the need for PCR and to send the samples to another lab, making it faster, easier to use, allowing for more samples at a cheaper cost, and can sequence longer DNA reads. Some challenges I have faced include fine-tuning my research question. This is a very exploratory project, so figuring out a specific question to try and answer has been difficult. Also, figuring out what type of analysis to do. The questions I'm looking at are based on degrees of difference between the groups I created, so we are unsure what analysis portion will look like. I've been unable to learn specifics on where bees are going. Because bees are in an open system, it is impossible to know exactly where they are going and coming into contact with. A lot of the information I've gathered is speculation. And lastly, publication bias. We know that the industry that there is a lot of components that determine whether papers get published. There is also a disconnect between science and the policies created. By talking to labs doing similar research, I hope to learn more information about their process and perhaps any information they did not include in their publications. Here's a list of my references. Thank you so much for listening.